Hello guys, I am Shubham from Pro Tutorials. In this video, we are discussing amino acids incorporation during translation. Okay, we know that tRNA जो है tRNA that brings specific amino acids, right? Specific amino acids, and amino acids when bind to tRNA, that is known as tRNA charging, right? tRNA charging means the process in which amino acid is attached to the tRNA. So that video we have discussed in detail this process, right? tRNA charging. उसी वीडियो में हमने लास्ट में एक पॉइंट देखा था, दैट इज ग्लूटामेट इनकॉर्पोरेशन. ओके, ग्लूटामेट इनकॉर्पोरेशन ये पॉइंट हमने देखा था. तो इस वीडियो में दूसरे अमीनो एसिड्स जो इंकॉर्पोरेट होते हैं दैट वी विल स्टडी. ओके, देयर आर स्पेशल स्ट्रेटजीज बेसिकली, okay, or we can say special scenarios that we have to discuss, and those are very important while studying the translation. मतलब अमीनो एसिड्स कुछ ऐसे अमीनो एसिड्स है जो अलग तरह से इंकॉर्पोरेट होते हैं. ओके, so first one is the glutamate. I will not be discussing glutamate here because we have discussed that in the tRNA charging video. So please watch that video before starting with this video. तो ग्लूटामेट इंकॉर्पोरेशन का उस वीडियो में हमने पूरा डिस्कस किया है. इन दिस वीडियो वी विल डिस्कसिंग द रेस्ट अदर अमीनो एसिड लाइक सेकंड सिमिलर अमीनो एसिड्स होते हैं लाइक आइसोल्यूसिन एंड वैलिन, ओके, एज वेल एज टायरोसिन and phenylalanine. These are similar amino acids. इनका incorporation कैसे होता है that we will see, as well as next is a unusual amino acid that is selenocysteine. Very important. This is the 21st amino acid. As well as there are some other modifications. That we will be discussing in this video. So this video is very important. Okay, apart from the translation process that we have discussed, initiation, elongation, termination in prokaryotes and eukaryotes, this video is important to the point for amino acid incorporation. मतलब amino acid का incorporation कैसे होता है. Okay, so there are special cases. The first case is glutamate. Again, I will repeat that please watch the tRNA charging video. I will attach in the I button. In that, we have discussed the glutamate case in detail. Okay, so that I will not repeat again. tRNA has 3' acceptor end, right? We know this is very important, and the anticodon part, anticodon loop. These two things are very important for the proper amino acid. Okay, correct amino acid को लाना है तो recognition के लिए 3' acceptor and anticodon are very important in tRNA structure, right? So now these are the similar amino acids. Similar amino acids. Okay, how they are incorporated, let's see. This is a tyrosine first of all and phenylalanine. Same structure है. Tyrosine में OH है, OH. This OH is important for the strong hydrogen bonding. Strong hydrogen bonding. स्ट्रॉन्ग हाइड्रोजन बॉन्डिंग होता है, which is why tyrosine is properly recognized because of this OH. Right? You have to remember this. The next case, isoleucine and valine. Isoleucine has this extra methyl, right? Extra methyl. So this is a large molecule basically. ये बड़ा molecule है compared to valine, right? So valine का जो tRNA है, वो valine को कैसे select करेगा? Because isoleucine is larger molecule, so this will not fit into the tRNA. Okay, tRNA catalytic pocket. Term है that is catalytic pocket. So catalytic pocket जो है tRNA का, valine tRNA का, उसमें isoleucine fit नहीं होगा because of its large size, because of this extra methyl group. So valine will be effectively selected. Okay, this is the first point. Okay, and when we are seeing that isoleucine tRNA में valine slip हो जाएगा. Okay, because valine is small, valine उसमें fit नहीं हो सकता in the isoleucine, right? So catalytic pocket is one important thing, and the next important thing is the editing pocket. Editing pocket. Some amino acids use editing pocket also. Editing pocket भी होता है. Okay. Editing pocket में क्या? Adenylation के products bind होते हैं. Right. Suppose in the case of valine, AMP valine will bind. AMP valine will bind and उसका होगा hydrolysis. Means this AMP will be released out and valine will become free then. Right, valine free होगा, AMP release होगा editing pocket में. Means or what we can say, editing pocket increases fidelity. Fidelity इसे और भी increase होती है. Fidelity means accuracy. Right, accuracy is increased. Right, so these are the things: catalytic pocket, editing pocket, valine and isoleucine के लिए. ओके, टायरोजीन को रिकॉग्नाइज करना है तो टायरोजीन टीआरएनए स्ट्रॉन्ग बाइंडिंग होती है OH के साथ, राइट? OH हाइड्रोजन बॉन्डिंग होती है स्ट्रॉन्ग, एंड thus the tyrosine is recognized properly, right? One more thing we will see here, that is difference between catalytic pocket and editing pocket. Catalytic pocket में adenylation for adenylation. Okay, मतलब catalytic, from the name only we can learn it is responsible for catalysis. Catalysis मतलब, see, both of these are present in tRNA synthetase. tRNA synthetase are important for tRNA charging, right? So adenylation के लिए catalytic pocket and editing pocket में क्या होता है? Products of adenylation. Products of adenylation. For example, हमने देखा भी in the case of valine, editing pocket में AMP valine bind होगा, which releases valine after hydrolysis, and AMP will be separated, valine will be separated, right? मतलब यहाँ पे products of adenylation bind होते हैं, and in the catalytic pocket adenylation ये होता है, right? So these are the two important things and differentiating factors, and both of these pockets are important for the valine and isoleucine, and also some other amino acid also, right? Like alanine. Okay, so now next point is ribosomes. Ribosomes have no role in correct recognition of amino acids. This is also a very important point we have to remember. अमाइनो एसिड रिकॉग्निशन करने के लिए राइबोसोम्स का कुछ रोल नहीं है, ओके, बिकॉज ये हमें कब पता चला, राइट, फ्रॉम अ थिंग, सी दिस इज अ tRNA for cysteine, right? When chemical reduction takes place, यहाँ पे alanine बाइंड होता है, राइट, alanine, यहाँ पे cysteine था. After chemical reduction, alanine binds and then alanine will get incorporated. Okay, right. So this will incorporate alanine in place of cysteine. Got it? This is an example from which we understood that ribosome recognizes tRNA. Okay, charged tRNA we can write. Okay, see, ribosomes recognize, क्या recognize करता है? Charged tRNA. ओके, बस देखेगा अमीनो एसिड अटैच है क्या टीआरएनए पर, इफ यस इनकॉर्पोरेट करेगा, ओके, बट नॉट द एक्यूरेसी, एक्यूरेसी नहीं चेक करेगा, ओके, लाइक नाउ वी सॉ सिस्टीन अटैच होना चाहिए था, सिस्टीन, बट एलानाइन है, देन टू द एलानाइन विल गेट इनकॉर्पोरेटेड, राइट? दिस इज द एग्जांपल ऑफ सिस्टीन एंड एलानाइन मॉडिफिकेशन. Now about the last thing, that is selenocysteine. Selenocysteine. Okay. This is the 21st amino acid and which is unusual. Unusual amino acid है. From the name only we can understand this is the cysteine. Okay. Cysteine है, चेंज हुआ है, राइट, सिस्टीन चेंज्ड, मॉडिफाइड. लेट्स सी स्ट्रक्चर. सी दिस इज सिस्टीन. सिस्टीन में एसएच होता है, सल्फर ग्रुप, यहाँ पे सिलीनियम है, सिलीनियम इन सेलेनोसिस्टीन. रेस्ट सेम स्ट्रक्चर, ओनली कंटेन सिलीनियम इंस्टेड ऑफ सल्फाइडल ग्रुप. So how is this incorporated now? Basically UGA. अभी ये तो स्टॉप कोडॉन है, इसीलिए तो हमें याद रखना है, राइट? दिस इज स्पेशल केस, स्पेशल केस है, कुछ तो स्पेशल सीक्वेंस है which is responsible for the selenocysteine incorporation at the stop codon UGA. Okay, very important. And this is incorporated by serine tRNA. Serine tRNA. Okay, and यहाँ पे chemical modification नहीं है, right? No chemical modification after translation वगैरह ऐसा नहीं है. This is done enzymatically. Enzymatically. Okay, special tRNA. Special serine tRNA that enzymatically changes. Okay, serine से conversion होता है selenocysteine, right? Next point, serine tRNA synthetase carry out this reaction. Okay, serine tRNA synthetase. See, first serine incorporate, right? Then serine is enzymatically modified, okay, by serine tRNA synthetase. Okay, and selenocysteine हमें कहाँ मिलता है? Catalytic center of enzymes. Catalytic center of enzymes में selenocysteine होता है. So this is the 21st unusual amino acid. Okay, and मतलब this is enzymatically modified, right? There is other one, just for example, hydroxyproline, which is also a unusual amino acid, but इसका chemical modification होने से ये incorporate होता है. Okay, this is found in collagen, and this is chemically modified. Modification. Right, due to chemical modification, hydroxyproline is incorporated. Got it? So this is just for an example, and this is all about amino acid incorporation, and all these things are very important while you are studying translation. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Thank you guys.